The overall goal of this procedure is to evaluate the in vitro suppressive function of regulatory T cells isolated from chronic virus-infected mice.
This method can help answer key questions about the phenotype and in vitro suppressive function of activated regulatory T cells during a chronic virus infection.
This technique can evaluate the direct effect of regulatory T cells on the inhibition of CD8-positive responder T cell response in vitro. This is a great advantage of this technique.
Begin by transferring 5 × 10 to the 5th total splenic lymphocytes in 50 microliters of FACS buffer into each well of a new U-bottom 96 well plate, followed by 150 microliters of FACS buffer. Centrifuge the plate and discard the supernatant. Then agitate the cell pellets and wash the cells 2 more times in 200 microliters of FACS buffer per well. After the second wash, resuspend the pellets in 50 microliters of antibody cocktail per well for 20 minutes in the dark at 4 degrees Celsius. Then wash the cells 2 times in FACS buffer and decant the supernatant. Next, fix the cells for 20 minutes in the dark at 4 degrees Celsius with 100 microliters of fixation buffer, followed by 2 washes with permeabilization wash buffer. After the second wash, resuspend the cells in 50 microliters of anti-FOXP3 antibody.